Hi, my name is Kelly Cowan, Marjorie Kelly Cowan, and I'm the author of two microbiology textbooks. One is called A Systems Approach, and the other one is Fundamentals of Microbiology, a Clinical Approach. And I would like to give a little bit of an update on vaccines and COVID. This is August of 2021. I recorded an earlier presentation, but things have changed as they do, and I wanted to give a bit of an update. I'm going to give background first and then go straight into the COVID vaccines. Figures that I'm going to be using in this presentation, if you're using the Fundamentals book, they'll be in Chapter 13. If you're using the Systems Approach book, they'll be in Chapter 15. So first, going over some very basic fundamental things about adaptive immunity, which is what vaccines use. There are three major things we're going to talk about today. First of all is specificity. The immune response, we call it specific or adaptive because it responds to an antigen, a piece of a microorganism that it has seen before and been sensitized to. And those parts of the immune system that have been sensitized to that will not recognize or respond to other antigens or other microbes, just the one they saw in the beginning. The next thing we want to talk about is inducibility, and that is that the adaptive immune response is generally in the off position, and it only is activated once the antigen to which it has become sensitized in the past appears again in the body, and then it's induced or turned on. Lastly, we'll talk about memory, which is really the cornerstone of adaptive immunity, meaning that once the immune system has been primed to recognize, let's say, the SARS-CoV-2 antigens, it remembers them in the sense that it has created a whole bunch of cells that will recognize it very quickly next time it appears. And we call that memory. So there are four types of adaptive immunity. This is all that very specific immunity I was talking about. There is active immunity is what we're most interested in for this topic. Now, active immunity either comes from having the infection and recovering from it, then you will have memory. Your immune cells will have memory to that infection and respond quickly and effectively next time it encounters it. We can also mimic the natural process of active immunity by exposing your immune system to antigens in such a way that they are what we call antigenic. They produce memory and immune response without producing disease. And we do that through the use of vaccines. That's the principle of all vaccines. Give the subject, the patient, antigens in a safe, non-disease causing form that still recruits all of these cells that are going to be specific and induced when the actual antigen is encountered in the environment. So active artificial immunity is basically the use of vaccines. Very quickly, I know this is review for you, but if you look at this chart, you will see the dark blue, navy blue line is an indication of the total amounts of antibody to a particular antigen in the bloodstream. That's what it means by antibody titer or concentration on the left. And you will see that the first time you're exposed to this antigen, either naturally or through a vaccine, there will be a period, a latent period of several days before you get significant amounts of antibodies in your bloodstream towards the end of what's called here the primary response. Generally, that latent period gives the microorganism, if it's a real live microorganism, time to actually cause disease. Now, of course, that doesn't happen when the first exposure to the antigen is a vaccine. What happens after we cross the white area and move into the light blue area, you are exposed for a second time to the antigen, then your response is very quick and much more robust if you look at the height of this navy blue line. And the whole point of vaccines is to artificially create the primary response with the yellow background so that you will have a robust secondary response as seen in the portion of the graph with the light blue background. Now, when the first exposure is a vaccine, as I just said, you will go through the primary response without having symptoms of the disease. In the case of the vaccines available for COVID-19, a booster shot is recommended, and it's given also sometime during the primary response. And as we've just heard, there's probably going to be a third shot, which is a second booster, also given. And it's all meant to just increase the efficacy of and the concentration of antibodies while there is no actual disease present. All right, let's talk about how vaccines are typically made. You have heard about these. These are the vaccines before COVID. These are all of the vaccines that you've ever had have been made this way. And there are three major mechanisms. First of all, at the top, you take a virus or you take a bacterial cell and you inactivate it or you kill it. And that is used as a vaccine injected into a person so that the antigenicity is retained, but it's dead and it can't cause disease. It stimulates immunity, but the pathogen cannot multiply. In another method that we use, number 2, the microorganisms are still alive, but the parts of them, the genes or outer surface proteins or something, have been changed, eliminated, or reduced so that they are still antigenic, they don't cause disease, and the advantage here is that they can continue to multiply and continue to boost stimulation to the immune response. An example of that is the MMR shot, measles, mumps, and rubella. And I forgot to say, but the example of the inactivated virus that is used as a vaccine are our flu shots. Okay, down to number 3, where we're just using parts of the microbes. We either harvest surface proteins from a virus or some part of a bacterium and use those parts in a vaccine. So the whole pathogen is not present, so it can't cause disease. Or more frequently, recently, is we determine the genetic sequence in a virus that creates a protein that will be antigenic, and we take those genetic parts, put them on a plasmid, and cause a completely unrelated microorganism to create copies of that protein, and we use that as a vaccine. So that's even one more step removed from having an actual organism there. The organism is not anywhere involved. We just copied the sequence of its DNA to create a protein that will be antigenic. And this is also the process used for diseases mediated by toxins, let's say tetanus, we take the toxin, either genetically engineered or from the organism itself, and we alter it chemically or genetically so that it no longer causes the symptoms, but it's recognized by the immune system. So the immune system can immediately shut down a toxin when it is released in our body from a microorganism. So examples of these are the hepatitis B vaccine and the tetanus vaccine. Okay, then enter the mRNA vaccines. mRNA vaccines seem new. They are new because these are the first ones that have ever been licensed for use in humans. But the technology has been around for at least 20 years, and mRNA vaccines have been researched, created, improved upon for years and years and years. And the way it works is, again, we find the sequence for the virus's spike protein. The spike protein is going to be the one that's very important for both attaching to human cells and also for our antibodies attaching to it to keep it from attaching. So we take the genetic sequence for the virus's spike protein and we create mRNA out of the genetic sequence. You remember, messenger RNA is the message, the script for transcribing RNA into actual proteins. So we take the mRNA script, it's packaged in a lipid nanoparticle. Lipids because then it will fuse with the lipid membranes of our cells and enter the cell quite easily. And once it's inside our cells, our cellular machinery uses the mRNA to transcribe proteins. And these proteins are displayed on the surface of our cells and stimulate an immune system response. Just as a little bit of an aside, I want to show you very briefly how vaccines are approved, studied and approved. And this is true for many kinds of drugs. Food and Drug Administration has the rules which say there have to be 3 phases completed, absolutely completed, no shortcuts, studying a new pharmaceutical like a vaccine before it can be even issued emergency use authorization as the COVID-19 vaccines have. And the phase 1 is with a few people just to see if it's safe in these few people, looking at dosing. Does it seem to have the desired effect? This is very early on in a handful of healthy volunteers. If it passes that test, then we move to several hundred volunteers in phase 2, asking the questions, what are the most common side effects? And are the volunteers' immune systems responding effectively to the vaccine? If it passes those tests, and sometimes phase 3 is started as phase 2 is coming to an end. Phase 3 involves hundreds, usually thousands, almost always thousands of volunteers asking the questions again. Is it safe even in all of these people? Is it effective in all of these people? And how do people who get the vaccine and people who do not get the vaccine compare in their natural experience when they're exposed to this virus? In that case, the FDA will license it. In the case of the COVID-19 vaccines, it was given emergency use authorization. It happened very fast. A lot of people are concerned about how fast it happened. And it did happen very fast. It can happen very fast if it has to. The trouble is there are so many drugs in the pipeline and so many other things in the process with the FDA that it takes a long time to get the studies conducted It's expensive to conduct the studies, all of the phase 1, phase 2, phase 3. There's a long line at the FDA to get approval to move on to other phases, et cetera, et cetera. But we were in the middle of a pandemic. So people who are well experienced in how to conduct these trials were able to put it as a priority. They got money from multiple government initiatives because we needed to get the majority of people in the world vaccinated. And so all of these processes were still followed. It's just they were put on these vaccines were put on the front burner. Just to be complete, if you look at the green box, once a vaccine is released and used on hundreds and thousands and millions of people, it is we call this phase 4 sometimes. It's a little misleading because the vaccine has already been released, but it's okay. And now that the vaccine is being used in so many people, we have a much finer ability to look for rare side effects and rare cases of it not working, et cetera, and to look at exactly how effective it is. No vaccine is 100% effective. It's not black and white. You have the vaccine, you're 100% protected. All vaccines have a percentage of effectiveness. 95 out of 100 people will be protected. 85 out of 100 people will be protected, et cetera, et cetera, et cetera. It turns out that COVID-19 vaccine has a very high effectiveness. Just a little walk down memory lane, or history lane as it might be. These vaccines for these diseases, pictured smallpox, polio, measles, rubella, etc., they were all developed and deployed in the 20th century. And I just want to show you how absolutely effective that they are. The percent reduction is the bottom line in bold. Smallpox was reduced by 100%. The numbers on the top are how many people per year in the 20th century, on average, had cases of smallpox. It was 50,000 in the U.S. per year. 100% reduction after the vaccine was introduced. Polio, the same thing. 100% reduction. Diphtheria, measles is 500,000 cases a year in the 1900s. And now we have just a handful, and that counts as a 99.99% reduction. Before these vaccines, any single one of these diseases could kill you. Okay, let's talk about what's on the market right now. What you've probably already received one of these, or maybe you haven't. But let's talk about that. We have 2 mRNA vaccines that are available: the Pfizer vaccine and the Moderna vaccine, which act the way we discussed earlier for mRNA vaccines. We also have the Johnson & Johnson vaccine, which is an adenovirus vector that contains a protein made through recombinant DNA technology, which that's been done forever and ever and ever. The hepatitis B vaccine, which you probably have received, is also created this way. Let's address the side effects of vaccines. So far, through August 9th, 2021, 351 million doses have been administered in the U.S. under the most intensive safety monitoring in history. And the side effects are, the rate of side effects is similar to the rate of side effects in all currently approved vaccines. You may have heard about the Vaccine Adverse Events Reporting System that I will abbreviate as VAERS as I talk about it. And this is a system set up by the government in the 1980s to give ordinary citizens, anyone who has received a vaccine, or their parents, or their healthcare provider, can report to this. It's now a website, of course, but can report if any symptoms occurred in the period following a vaccination. So you will hear news outlets talk about the reporting system and how many side effects have been registered there. But what you need to know is that none of these have to be verified. Anybody can register a concern. I had a severe headache 3 days after the vaccine, or I got the flu after I got the influenza vaccine, which is not possible, by the way, or my toe really hurts and I had a vaccine a week ago. All of those things could be entered into the reporting system because it is designed to collect every possible situation, and many of them will be found not to have been caused by the vaccine. But the system was designed to collect every piece of information that can later be used for research, and that patients can also have an outlet to register their concerns. So if the VAERS says that there have been, that 3% of vaccine recipients have complained of the following complaint, that's just correlation. It's not causation, and it hasn't been shown that was due to the vaccine. Also, something you should be aware of is that there are websites that have been set up that are meant to mimic official government websites like the CDC, like the Vaccine Adverse Events Reporting System, and they often contain inaccurate data. And so it really is important that you find out, look at the source of the web page that you're looking at to determine whether it's trustworthy or not. The summary is, though, that overall the significant side effects affect 1 or 2 people out of 1 million doses given, which is in keeping with most of the other vaccines that we are administered throughout our lifetime. Here's a screenshot of the official Vaccine Adverse Event Reporting System, and there's a paragraph there about important things to know about VAERS, and I encourage you to take a look at that and read the important things to know. All right, let's very briefly talk about herd immunity that you have heard about in the United States and elsewhere. And it means that enough of the population has been vaccinated with a highly effective vaccine so that transmission stops because there are not enough susceptible hosts. In practice, what does that actually look like? And because herd immunity is kind of a funny phrase, I decided to use a herd of bison to illustrate this. If you will follow me and look at the green star, that bison is carrying a virus that I'm going to call the bison virus. And if none of the other bison were masked or vaccinated or had the disease and recovered from it, then that bison virus, beginning near the center of that herd, would spread quickly through all of the susceptible bison around that particular one. But in this example, the bison with blue X's have been vaccinated, and the ones with the red X's are observing good mask hygiene, are wearing their masks. And what I don't show here is that there are probably other bison who have had the disease and recovered from it, which further reduces the percentage of susceptible individuals in that herd. Now, of course, humans are modeled exactly the same way. And what we have been told to this point is that about 70% herd immunity, 70% vaccination will get us herd immunity, meaning only 70% of the population needs to be fully vaccinated. That number, the percentage of the population to reach herd immunity, is different for every different microorganism. For instance, measles is notoriously difficult. We really have to have 95+% of the population vaccinated against measles because it's highly contagious, and anything lower than that will not achieve herd immunity. But as far as we know, at this point, 70% of the population being vaccinated should provide effective herd immunity. Now, what we know in the U.S., and this is mid-August, currently the percentage is 51%, not close enough to get herd immunity. And of course, infectious diseases are local, and the different states range from having a 36% vaccination rate, very low rate, and some of them reach 68%, which is getting close to herd immunity. But of course, people travel. We really need the entire country to be averaging around 70%. Now, what you have seen is that variants arise, particularly when a long period of time goes when a virus is able to multiply in immunocompromised people who aren't vaccinated. That gives viruses more time to mutate, which inside a host, because they're not going to do anything outside of a host. But when they're inside a host, that's when they are going to mutate and give rise to variants. The variant we're worried about currently, of course, I say of course, in a month it could be something different. It's the Delta variant. And I want to explain to you how the variants work and what they mean for infection and transmission. So viruses, this little green circle is attaching to the human cell, and the virus green circle has spikes on the outside, and it uses those spikes to attach to the human cell. And where the red diamond is, those are amino acids in the protein sequence of the spike that are used to attach specifically to human cells. And if you look on the large blown up spike there's also an area on the spike which is recognized by antibodies that are manufactured in response either to a vaccine or to actual infection. And those are usually in two different spots on the spike. They might not be very far apart as illustrated here, but they are distinct. And the infection that taught us about how these spike variations work is influenza. You know that you have to get a new influenza vaccine every year, and the reason for that is because it is constantly mutating, and eventually one of the mutations is going to overcome the rest of the mutations. And it has been shown that with influenza virus, the amino acids that bind to the human cell, the red diamonds, they're not really mutating at a very high rate at all. So the influenza virus can continue to bind to human cells the way it always has, which you can see is advantageous to the virus. With influenza, it is quickly mutating the amino acids in the part of the protein spike that are recognized by our antibodies. So that's very clever, and over many, many, many years of mutations and evolution of the virus, it has found a very clever mechanism to maintain its binding to the human cell, but to change the amino acids that our defenses will recognize so that it can escape the defenses. So every year, scientists analyze how those blue amino acids have changed in the dominant strains of the virus, and that's what they make the new vaccine out of. Now, the Delta variant does something different, and that's probably because it's brand new variant. It hasn't evolved over generations and generations and generations of humans and or viruses, really. And we find that the vaccine still protects you from the worst consequences of infection with Delta, but that even fully vaccinated people are still infected with Delta. And the science is not sure about this yet. We're still investigating it, but one possible explanation is that the area on the spike of the Delta variant that is recognized by human antibodies has not changed. And so our antibodies are still effective at preventing severe disease and death. However, the area of the protein spike that binds to the human cell is changing. And in this case, it's caused the variant to bind more tightly, more effectively, and that leads to increased viral loads in the upper respiratory tract and increased transmission. So let's think about this. This is in a person who's fully vaccinated. They themselves are not going to get severely ill, but they can pass the virus on to other people. Well, here we have to think about the different classes of antibody. The virus, the Delta variant, is binding to human cells very tightly. And even though the antibodies are still effective against it, the best antibodies to prevent colonization in the upper respiratory tract, which is mucosal membranes, of course, is the class of antibody IgA, secretory antibody, that is secreted onto mucosal surfaces, and if it were present there, it could prevent the attachment, the survival of the Delta variant. But most injected vaccines are not very good at raising the class of antibodies that we call IgA. And so one hypothesis is that the virus does attach, is relatively unbothered in the upper respiratory tract by your antibodies of the IgG class, which are systemic throughout your body. But that the reason it doesn't invade and cause deep disease is because the IgG antibodies prevent it from doing that. Of course, new variants can be created every day and are being created. And the right combination of mutations in the protein spike and elsewhere in the virus will determine whether the new variant will become dominant or not. I'd like to sum up by saying that the vaccines do protect from severe disease and death amazingly well. And in fact, in terms of effectiveness, the percentage of the population who is vaccinated who will in fact be protected, this vaccine for COVID-19 has higher effectiveness than many of our current vaccines. And as if to emphasize that, I want to show you this graph of various states in the United States and who is getting disease now. And if you look at the right-hand column with the orange bars, over 90% of people who are getting the disease are unvaccinated. and the green bars indicate those who are fully vaccinated and what percentage of disease they represent. Not all 50 states are here and in this graph, and that is because the organization that did the research, KFF, used only data from states that have rigorous reporting practices and a compliance protocol that was considered useful to inform the analysis. Okay, as I said, the vaccines protect amazingly well, even with the Delta variant. They have higher effectiveness and fewer side effects than most childhood vaccines. And I am going to provide you 3 non-governmental sites that provide reliable and accurate information, both about the statistics of who's getting the virus, the biology of the virus, almost anything you'd want to know. Johns Hopkins is probably in the forefront of this effort, and that's their website. Forbes magazine is considered to be extremely reliable and scientifically accurate, and they have compiled all of their information and stories about coronavirus on a single page. And then there's an organization supported by many universities and scientists, and the organization is called endcoronavirus.org. I give you these sources because, for better or for worse, some people have become suspicious of government websites, and despite the fact that the information that the CDC and the WHO provide is accurate, I wanted to provide those of you who are skeptical with the opportunity to look at other completely vetted, scientifically reviewed sites for you to get your information on coronavirus. So that's the summary for August 2021. I wish you well, stay safe, and I hope your studies go well also. Thanks.